I know this paper was really complicated, but my own personal opinion, if you are going to learn something like genetic engineering, learn about proteins and stuff like that, there's no easy way to do it. You just kind of have to like throw yourself into the fire, drink from the fire hose, and hope you survive. You failed the bioethics certification? That's funny. You think that was funny? You guys like that? That was a pretty funny website I made. But today we're going to talk about the dynamic personalities of proteins. Talk about proteins and how they function. If everybody's got their chat set to live and not top, I want to post this link. So with the link I was trying to post last time, the RCSB, that's where they have all the structures of proteins. Now, if you want to understand how proteins work and you want to look at the structures and things like that, this is the place to start. And this is where we're going to start. We're going to understand basically just what proteins are, how we can look at them and understand them. So there are two main ways that scientists have found, mainly, there's maybe like three now that scientists have found in order to what they call solve the structure of a protein. We don't really say like look at, I don't know why. In general, when you're talking about protein structures, you say they solve the structure of the protein, because actually though cryo-EM, which is a new up and coming structure solving tool, you can actually look at the structure of proteins technically, but techniques like NMR and X-ray crystallography, which you might have read about in the paper, are two ways in which you solve the structure of the protein. So you don't actually get to see it. What you're doing is you're measuring things indirectly, and then from those things, you're rebuilding the structure of the protein. So yeah, Craig, it is basically a puzzle. Now, with X-ray diffraction, what they actually do is you purify a bunch of the protein, and then what you try to do is you try to grow protein crystals. It sounds like something out of a Harry Potter movie, right? One of the Horcruxes, protein crystals, maybe. Did I get that right? Maybe Pokemon. They got put protein crystals in Pokemon. Oh, they do. Protein potions. And what they actually do is they literally take concentrated protein and put them in little drops, and they let them sit. Have you ever like what you should do if you've never done it is pretty cool. Take some water and add a bunch of salt to it. Warm up the water, add a bunch of salt, and then just let it dry. Over time, what happens is you'll see salt crystals form, like big, nice crystals of salt, right? Because what happens is, the salt comes out of the solution and forms these crystals. Well, the same thing can happen with proteins, right? So proteins can form these crystals, which are just repeating patterns of the protein inside some crystal structure. It's pretty much trial and error. So some scientists spend their whole PhD, just literally setting up these crystal trays, right? Just keep pipetting protein and trying to set up these crystal trays. There's even robots that do this for people. So they'll just use a robot that'll set up these crystal trays for them. Once you have this protein crystal, what you can do is you can shoot X-rays at it. It's like pew, pew. You shoot X-rays at it, and the X-rays will diffract off this crystal. And then you see how they diffract off. And what you can do is you can take that data, diffraction data, and from it, you can actually solve the structure of the proteins, right? Because based on the the diffraction pattern, you can find out where the nuclei are and recreate this protein structure. Now, this takes pretty strong X-rays. So generally it's done at like places, national labs called synchrotrons. These, you can look them up. They're these giant facilities that get measure all types of physics stuff and are able to shoot these X-rays. X-ray crystallography is one of the main ways that protein structures are solved, right? So we can't usually actually see proteins because they're so tiny, but we reverse engineer and solve it. The other technique that they talk about a lot in this paper is called NMR, nuclear magnetic resonance. It's what I studied, what I used a lot during my PhD. NMR is this crazy technique. It's super crazy. So the way NMR works is, what they found is that if you take a giant magnet and you put any sort of compound inside it, whether it's water or whatever, some sort of molecules inside it, what you can do is you can shoot a high-powered radio frequency pulses at these atoms and nuclei, and these high-powered radio frequency pulses will cause these nuclei to basically excite, right? And when that happens, you can measure this excitation and decay of the nuclei. It's way more complicated than that, super complicated. But what you basically get is they make these giant superconducting magnets that are cooled by liquid helium, right? Because liquid helium, superconductors only operate at very low temperature. So you charge up these giant magnets, poof, right? And you keep them cool in liquid helium, and then you use liquid nitrogen to keep the liquid helium cool, right? So you need like 2 layers. And from this, what you can do is you could stick proteins in there. Generally, you stick in proteins that have labeled atomic nuclei. And by that, I mean, generally, for instance, the predominant nuclei for carbon is carbon 12, right? And for nitrogen is nitrogen 14. But what you can do is you can grow up like bacteria or something with no nitrogen except for N15 or C13. And when you do that, all the protein molecules have these different nuclei that have extra characteristics. These nuclei, they're spin polar, they're easier to excite and see in this process. From this, now what you can do is you can excite all these different nuclei in different ways, right? So you can excite all these different nuclei, measure how they affect each other and all this different stuff. And from this process, what you can do is you can recreate protein structures. So you can solve the protein structure. Now, NMR is way more complicated to do than X-ray crystallography, but it also is a commonly used technique. So if you're looking at the RCSB or something like that, and you're looking at protein structures, generally they'll say how the protein structure will solve. If it's a crystal structure, if it's an NMR structure, if you use X-ray diffraction. And those are the 2 main techniques. Now, there's started to be another technique that's coming around, and it's called cryo-EM. So maybe you've heard of electron microscopy. Well, what this does is it uses super cool electron microscopy. And if you put a bunch of protein on like a surface, stick it in the electron microscope, there's enough protein on there that you can use image software to rebuild the structure of protein based on all the different protein molecules that have adhered to the surface. So you can imagine the protein molecule adheres to the surface. Some of them are going to be sideways, some of them are going to be up and down. They're going to be all different angles. Well, using modern image recognition and rebuilding software, you could rebuild this structure. So cryo-EM is starting to be a faster, better way. Generally, you need larger structures for cryo-EM, but I think they're getting pretty good at it now to where you don't necessarily need that, right? So going into this paper, we have to understand, right? This is how we look at proteins. We can't look at proteins with our eyes, really. So we look at them using all these crazy fancy techniques, right? No, generally larger structures, Christopher, have lower resolution because there's so many different things going on. So you can imagine if I take a picture of, say, a black and white image with no color or anything like that, as compared to a color image, or a color image that has all different shadings and things like that. It'll be easier to extract like lines and things like that from the black and white image than from the color image, because the color image is more complex, it's more complicated. The black and white image is simpler. So larger structures tend to be more complicated and so generally have lower resolution. You also have to understand that a larger structure, when you're looking at it in, say, X-ray crystallography, there's more freedom. There's more degrees of freedom. If you have a 50 amino acid protein structure, the degrees of freedom are a lot smaller than if you have a 1,000 amino acid protein structure, right? There's going to be a lot of different places where things can be different, move around different, look a little different. But yeah, so this paper, it mainly focuses on NMR, but it does talk about X-ray crystallography a bit. Now, here's the crazy thing about proteins, right? Proteins aren't just like a static thing. They're actually moving around constantly. And they're not only moving around constantly, they're moving around on different time scales, right? So you can think of a protein kind of like a car or something like that. In a car, you have the wheels spinning at a certain RPM. You have the engine moving at a certain time RPM time scale, right? You have you yourself turning the wheel at a certain time scale. You have all these different things going on at different time scales that affects the function of the car that you're driving, right? All these different things. Now, if you were to try to understand the car from scratch, say you had no idea what a car did or how it functioned, the first thing you try to look at is what things move at what time scale, because then you get an idea of what's moving together, right? So if all the wheels are spinning at the same speed, you could probably have a good idea that the wheels are all doing something together, right? They're all spinning at the same speed. Why are they spinning at the same speed if they weren't doing something in concert together? You can think about that across all the different time scales. So proteins are moving at many different time scales as talked about in here, right? There's like the nanosecond time scale, the micro to millisecond time scale, all these different time scales. And these time scales, motions, affect how the protein function. So proteins, they are not only this structure, complex structure of amino acids, they are a machine, a complex moving structure. And that makes them even more crazy, right? Now, the big thing about proteins, as we talked a little bit about last time, is that we can't really understand proteins by looking at a single protein. Just like we couldn't necessarily understand a car or a cell phone by looking at one of them if we didn't know what it was. you'd have to look at a lot of them and figure out what they have in common, what's going on. And this is what statistical mechanics is in thermodynamics, right? So statistical mechanics and thermodynamics, what they allow us to do is look at massive amounts of something, right? Like a lot of protein. If you like extract protein from bacteria, you'll have a lot of it, billions of molecules. And from these, we can understand all the different states, all the different interactions that proteins have, right? That's the cool thing. Because you're looking at a billion of them doing something at once, right? You're looking at a billion of them being a protein. And from that ensemble, from all those different proteins, you get a whole wealth of information. Now, generally, the state that a protein is in, right, they call its ground state, or its, normal state, with the state at its lowest energy conformation, because a protein does exist in a certain, normal state. It has a function. Like if it's an enzyme and this enzyme breaks down sugar, breaks down glucose, that's its normal function. Now, it can exist in other states for sure, but those states are controlled by, as it talks about in paper, the thermodynamic equilibrium. Now, what is that? I know it sounds like nonsense. Basically, the thermodynamic equilibrium is if we have a, a constant temperature, pressure, and other things. Generally, with proteins, we focus on temperature, though pressure can be a thing in like deep sea fish. Generally, we focus on temperature with proteins. If you're at a constant temperature, these proteins should generally, in a distribution, in like a normal distribution, a Boltzmann distribution, they call it in thermodynamics, they should have a function, and then they should have a bunch of other states in that are on the tails of that normal distribution. Now, these other states can be populated by shifting this temperature, right? So if you have a state that takes a lot of energy to get to, right? So normally a protein is in a shape. It's in a specific shape. If you want your protein to just be a straight string, right? Completely unfolded in no state, that's going to take a lot of energy, right? Because you have to fight against all those interactions that want to keep it folded. So you have to heat it up really hot, right? It takes a lot of energy. Heat is kind of like relational to energy. Not just heat, but you can also use different things like chemicals, right? Certain chemicals, guanidinium hydrochloride, urea, other things, what they do is they will unfold proteins so they have no structure. It's a common technique used to study protein folding and understand things like that. So there's these differences in energies, right, between the state the protein wants to be in and another state it could possibly be in. Now, there are other things that can do this, not just heat, right? I'm sure you've heard of like ATP and things like that. Now, ATP and things like that, what they can also do is they can also use energy, right? The breaking of a chemical bond, that energy that's released through ATP hydrolysis can power something. Think of it kind of like when you're snapping a toothpick or something like that, right? Or like a chopstick. When you snap it, right, the energy that you're putting into there will sometimes like shoot pieces of wood out, right? As the energy gets released from you breaking it. Think of a similar thing when you're thinking of like the bonds and ATP being broken and energy being released, right? So you're taking this energy that's stored and releasing it into the protein, which causes stuff to happen. In the cases of ATPases, these proteins actually rotate, right? Really cool. They move around. Um, let me see if I could find. There's actually a really cool ATP ATPase video. ATP synthase. Let's see, is this one that? Not really. How about this one? They're all so long. All right, look at this video. What you could see from this video is you can actually see synthase protein and how it rotates and how it moves, how it uses ATP to actually move, use the energy to change the structure, right? So that's what proteins are constantly doing, is they're taking energy on this very tiny level and using it to change the structure. One of the experiments we'll be doing in this class, I think in month 3, involves a protein. It takes light and it uses the light energy. So energy from a single photon, it captures that energy and uses it to actually move and change. That's pretty cool and crazy. So these are like they're nanotechnological light sensors, right? They're little nanotechnology that actually changes and moves in response to a single photon of light, blue light in the case of the protein. All right, so they talk in the paper a lot about different techniques on how to measure these different things. Now, those things are kind of important, but are not so important. Basically, what these techniques are telling you is just what is available to a scientist and what a scientist can possibly do, right? Like, what can a scientist actually look at in proteins? And if you look at, what is it, figure 1B, they talk about the different techniques that allow scientists to look at the different time scales of motion of proteins, right? And they talk about two different motions, local flexibility and collective motions. So if you think about local flexibility, that's like, you know, my muscles contracting and moving. And collective motions would be like all the muscles in my body moving to like throw a ball, right? So proteins have different scales of movement like that, just like human beings. So they have local movements, local flexibility that doesn't affect the whole overall motion of the protein. And then they have motions that can affect everything, where big parts of the protein come together for large scale motion. And those are generally the important thing. The faster scale stuff can be important, but it's not as important. Um, Christopher, I don't know. I don't know if people are using proteins for optical computing. Actually, proteins aren't that good of a substrate, right? Because here's what you have to think about. The reason computers work so well is because even when the temperature changes, everything still functions, right? But proteins and DNA, when the temperature changes, they don't function, right? Or they start to change function because they change states. If you heat up the protein too much, if it's like inside a computer and you heat it up too much, the protein dies. It doesn't function anymore. It unfolds, right? So using proteins in like the everyday life isn't it's an interesting problem to be able to solve. Uh, Are any of these techniques cost effective enough for biohackers to use regularly? So there are some techniques that you can use. Generally, these revolve around absorbance spectroscopies. So what you can do is both using fluorescence or just using absorbance, you can measure different aspects of how a protein's changing, right? Because protein, the amino acid molecules, absorb different wavelengths of light because of the way the electrons are arranged in some of the molecules, right? So generally, sorry, I'm just thirsty, I think. Darren, that's a really good question. Now, actually, I think Nobel Prize was won. Who won the Nobel Prize for RNase folding? 19, uh, 1972 Nobel Prize in chemistry. Was it Chris Amphison? I didn't think it was Amphison. Um, anyway, you can actually refold proteins. That's what's interesting. Not all of them, right? Not all of them. But you can refold some proteins. So some proteins, when you heat them up and then you cool them down, they will refold back to their normal structure. But understand that generally when you heat proteins up, sometimes what happens is they aggregate, like a cooked egg, right? Like a cooked egg, you're not going to be able to return it to a normal egg, at least not in any reasonable way possible. You could maybe return some of the proteins to normal by subjecting them to a lot of different chemicals and other stuff. But if you cool down an egg, as we know, it doesn't all return to like liquid. It stays solid. That's because all the proteins, not only did they unfold, but they start to interact with each other unfolded. And when they start to interact with each other unfolded is they call aggregate, right? So they start aggregating. They create these messes of proteins that are very difficult to separate from each other. Orthelin. We could talk about that some other time. I appreciate your questions. Hit me up on Facebook or something. We're trying to focus on protein folding right now. Protein folding. I know it's not that exciting, but it's really exciting to me. Super exciting. Now, one of the coolest things, I think, and understand proteins are like little nanotechnology, is using NMR. One of the things that they measure in NMR, they call it relaxation, NMR relaxation. And basically what that is a measurement of is what you can do is you can excite nuclei, right? So you excite an atomic nuclei. Now what happens is based on how much that nuclei is moving and interacting with things, it relaxes faster. So, right, if you heat up ice, if you make the molecules move around really fast, what happens is the ice melts, right? And if you heat it up even more, it turns into a gas. You have these different timescale motions that create different things. In proteins, the same thing happens. If you excite a nuclei, it'll kind of cool off faster if there's a lot of stuff it's bumping into. Actually, that analogy made no sense, but we'll just keep going with it. So it like gives its energy off to all the other things, right? So if it bumps into this water molecule, if it bumps into this other atom, right? If it's near all these other atoms, it's releasing its energy whenever it interacts with other things. And they say it relaxes, it relaxes. So what you can do is you can measure the time scale of motions in a protein by seeing how fast these different atoms relax. You can see which atoms in a protein correlate with each other in terms of relaxation. And then from there, you can uncover how this nanomolecular machine works, like what amino acids are important, right? because sometimes in proteins, there's just a few amino acids that are essential. And the rest of the amino acids kind of provide the scaffold, the structure for the rest of it. Just like a car, right? With a car, you can take off the whole body of the car and the car can still run just fine. You could take out the seats of the car could run just fine. Those things aren't essential. They're kind of the scaffold that allow the car to be used and work. Proteins are very similar. They have an overall scaffold structure, and then they have specific regions that are more important. All right, what's next? Laue, X-ray, man, there's so many crazy, complicated stuff in here. Does anybody have any questions? Any questions you want to ask? I think we can talk about computational simulations. I think we talked about a little bit about that last time. Nothing? A dance on many timescales. Yeah, no. It's actually so cool. It's so cool because we don't think about this on a large timescale because everything on like a big timescale we build, right? So we've never had to like solve what makes a technology work. But you have to kind of think of like it is an alien technology, right? A protein is this alien nanotechnology that we're trying to figure out how it works when we don't have the instruction book for how it works. And this is how we do it. Yeah, so absorbance spectroscopy. So usually absorption is correlated with electron density of things. So things that are more electron dense absorb things better. Now, in proteins, there are certain amino acids that are more electron dense than others, right? These amino acids tend to be tryptophan. You know, tryptophan is this huge ring structure. It has multiple rings in it, and it's pretty big. Now, these rings, what they do is they create delocalization of electrons into the ring. So these rings are very electron dense. You also have things like tyrosine and histidine, right? And so what you can do is you can actually measure the changes in the absorbance of these amino acids, right? As you say, like unfold the protein. So if you stick a protein in your absorbance spectrophotometer, this would be a really cool experiment. Maybe we'll figure it out for next class. You can take a protein, and what you can do is you can just heat it up and measure the absorbance every change in degree, right? Every degree change. And what you can see is you'll see a change in the absorbance over temperature, which tells you that the structure of the protein is changing, right? Because the structure, the absorbance of the molecule changes when it interacts with more or less things, right? So if it's completely open interacting with water, the absorbance is going to be different if it's packed tightly against the protein. So the absorbance changes, and you can look up. It's pretty easy. Look up the absorbance wavelengths for these different amino acids. So tyrosine and tryptophan are generally at like 280 nanometers. I think histidine's at like 215, but don't quote me on that. They're really easy to look up. If a cell is deprived of amino acids, you will just die. It will die. So a cell, the two things, Shiv, that cells need, remember, are like a nitrogen source and a carbon source, right? So the nitrogen helps build the amino acids, and the carbon helps build the amino acids and DNA and everything like that. So basically, if you want to grow any organism, give it glucose and some sort of protein source. Generally, people use yeast extract, tryptophan, things like that. I mean, tryptone, not tryptophan. And those you can make most any organism grow on. But yeah, you can't grow. Amino acids are essential to all forms of life that we know, because all forms of life are based on proteins, and proteins are based on amino acids, right? So now we'll talk about computational methods, right? Computational methods. Now, what's really cool is you can actually download, oh, one amino acid out of many. Well, some amino acids aren't necessarily essential because the cell can build them from scratch. And you can, it's actually, so let's say we delete every lysine or leucine amino acid from um, the genome. Generally, there are some amino acids that are considered more important than others, right? So amino acids like alanine, leucine, isoleucine, valine, these are considered hydrophobic amino acids. Especially when it comes to leucine and isoleucine, they're very similar in structure, so much that they're pretty much replaceable. Same kind of with valine and alanine. Now, this isn't 100% true, but it's generally considered something that you could do. You could take a protein, replace the valines with alanines, and not likely see an effect. Now, there are other amino acids like glutamic acid, aspartic acid, lysine, uh, amino acids that have a charge. Now, these charged amino acids, they're what forms these interactions, long range interactions in the protein, and also interacts with things like DNA, right? If you're going to interact with DNA, you want positively charged amino acids because DNA is negatively charged. So these positively charged amino acids, if you remove them, you're not going to be able to interact with DNA, right? So there are amino acids that, are a little more essential than other ones. But, we have all 20, and there's actually a bunch of interesting papers on this that have shown that, like, you can get away, I think, with like keeping the structure and function of proteins with like 5 amino acids or something. It's they're pretty interesting. All right, molecular dynamics simulations. So there's actually some software. There's both NAMD. Here, there's two pieces of software that if you ever want to simulate NAMD and GROMACS, um, you can download the software and find tutorials online if you actually wanted to try to simulate a protein. Uh, Protein simulations are super fun. They're super cool. Uh, it's, computationally looking at how these nanomolecular machines work. It's really interesting because you actually get to watch it as a video, right? In real life, you don't get to see what the atoms in the protein are doing. But in the case of simulation, you can actually see what the individual atoms are doing, and you can measure all different sorts of things about what's going on. And it lets you find out really interesting and cool stuff. In one of the papers I published, um, why when body temperature changes? Yeah, we don't really change. So you have to understand that, like, your proteins in your cells are what we call buffered, right? So your cells aren't just liquid water or something like that. It's a complex environment in which the proteins live. So there's all these different things that are buffering the proteins that are helping keep them in their ground state, in their normal state. So when your body temperature changes or something like that changes, that's what keeps your body still functioning and working together. But stuff does happen, right? Right? So when your body temperature cools down, the function of proteins slows, function of cells slows. It's just what happens. Um, Probably not enough to notice. Maybe I don't know if anybody's ever measured it. Maya, what is homeostasis? So homeostasis is this idea that there is a constant equilibrium that your cells and your body and proteins are, all these things are in. It's this state of normalcy, so to speak. And your body is constantly trying to fight to keep this normalcy going, right? No matter what you do, it's fighting to keep you alive, to keep your cells functioning, keep your proteins functioning, and all these different stuff. So, like, for instance, if you take a cell and you put it in a solution of salt, a high solution of salt, and all these salt ions are trying to rush in the cell is trying to push out these salt ions. It's trying to take in water in order to, achieve the proper homeostasis to keep it going. Darren, yeah. So the your epilepsy, I don't know if you've actually, um, if it's genetic. If the epilepsy is genetic, sequencing yourself, you could perhaps find information on the genetic and protein differences that would cause it. It is something that is totally possible to do. Um, and it could provide information. I'm not very familiar with epilepsy or genetic epilepsy and what might cause it, so I can't speak specifically, but it is something that seems totally reasonable to be able to do. So yeah, I encourage you to download, try out NAMD or GROMAX. I think we have a computational, maybe protein computational science tutorial. Let's see. There's probably a lot better ones. But here is one that you can look at if you just want to go through, use VMD, use NAMD and stuff like that. Um, and mess around with it and see what it's like to, look at a protein and understand how it works. So I think in this little paper I wrote, it's looking at Ebola protein and how Ebola proteins function by using VMD, something I told you, using the NCBI to look up protein information. It goes through a lot of different things on how to use computational biology to understand proteins. So generally, as a biohacker DIY scientist, you're not going to have access to NMR or X-ray crystallography. These are things that probably won't for a long time ever be biohacker available. NAMD, I don't know if it's a separate download from VMD. I think sometimes they come together, sometimes not, Christopher. So you'll just have to check it out and see. But yeah, no. So you're pretty limited in understanding the protein structure and folding, but that's okay because there's so many resources out there already about this stuff. And generally, if you're trying to do something like mutate proteins, what you can do is you can look at the protein structure that somebody already solved, and you can base your work off of that, right? And so you don't really need to do too much in terms of structure solving yourself. All right. Do we have any questions about this? The dynamic personalities of proteins? Very dynamic. I know, right, Craig? We are. We're standing on the shoulders of giants. Good thing. Makes the work easier, but that's great, right? All right. So before we finish up tonight, Esther's going to go over some stuff. Esther's around. She's going to talk you all through some stuff. Christopher, they're not necessarily time dependent. They're not necessarily time dependent. They are, but they're on a fast time scale. So you're talking micro to milliseconds. Proteins fold from like seconds to minutes, but generally the protein things that happen are happening on the micro to millisecond time scale. So they are time dependent, but we more focus less on the time dependence and more on what they actually do. They don't have cyclical nature as we think of it. I don't know what that means. I don't know what I think of cyclical nature, so hit me up, Christopher. Post in the Facebook group or hit me up on Facebook and I'll try to answer your questions more specifically. But right now, I'm going to pass it off to Esther and she's going to go.
Hey. Hey, hey, hey. Friends. You guys see me on the main page? Yeah, we're good? We're good. We're good. All right. So month 2. Hey. Hey, what's up, Abraham? Hey, Craig. Yeah, so month 2, we sent out a lot of month 2 packages this month. What? All the kids. All the children. Oh. Yeah.
All gone. Sent.
Yeah. Yeah, we sent them. Yeah. Today I'm going to show you how to do streaking of bacteria, how to streak bacteria and yeast. It's really important. It's because we only send you like 4 stabs. And this is for all lots of experiments that we're going to be doing in the future. So all the bacteria and organisms are usually occurring in nature and they don't harm humans, the ones that we send you. So we send you things like E. coli, like yeast, like brewing yeast, all that kind of good stuff. So yeah, I'll just teach you how to streak these things out. So I'm going to make a really large point to make sure you open all of the bags and carefully read all the instructions for each item because they all have different things and different storage temperatures. Generally, you can throw things like in the freezer, things that say -20 needs to be frozen, -20°C needs to be frozen. It's in the freezer. If it's -4, needs to be in the fridge. Usually like bacteria and like living things can go in the fridge, like in just like the regular temperature where you keep your butter and stuff. But DNA and things like plasmids need to be inside of the freezer, which is storing next to your meats. And that's where we store our DNA, next to our meats, our frozen meats. All right. So, yeah. I will show you. We have these little things. They're called stabs. Don't ask me why we call them stabs. I guess because we stab it. What it is, it's just a little bit of agar. And it's a really nice way to transport just like little tiny amounts of bacteria. And what we did is we took a little inoculation loop and we just literally swirled a little bit of bacteria and then stabbed the agar. Let's see. So yeah, let me show you how to quickly do it. You guys did a really great job with your plates last month. I took a look at every single one of them that got posted on Facebook, and they were pretty awesome. So good job, you guys. I'm very proud. I'm so proud. Yeah, so we're gonna take a plate that you've already made, and this is currently LB amp. And you see I labeled it. And yeah, so make sure when you take out all your plates to see if there's anything that's growing inside of it, like something fuzzy or like a little black molds, like anything that can potentially contaminate your strains, which can be really, really terrible because then you won't have a pure culture. And when you're doing your experiments, you could completely botch up everything that you're doing. So make sure you take it out of the refrigerator, let it kind of like, come to room temperature, and then just kind of like look at it to see if there's anything growing on it first. And if it looks very clear, I think you're good to go. Okay, so number one, put gloves on. As we learned in our sterile technique section, always put the gloves on. And then we take one of these loops that we sent you, these inoculation loops. I guess you can't really see them. It's just a white block. But you take one of them and you sterilely pull it out. There's a small end and there's a big end. It doesn't really matter what you use, honestly. But I'll use the big end just to kind of visibly show you what's going on. So you take the little E. coli and carefully unscrew it. And you literally just take the loop. No, Eric, size does not matter in this case. Lucky for you, right? I have chill. You stab it, and then you pull it out really slowly. And then you take your plate, and you gently streak it like this. Yeah. Discard. I know, it's like the worst bubble wand ever, Matthew. It's because like you can't blow bubbles in it. It's so useless. Okay, so there are a couple ways to streak bacteria. So what I did there is just like, a really quick and easy way just to get a stab, like a bacteria from a stab to a plate. The real pros, I'll show you what the real pros do. So I'm going to visualize the bacteria with a Sharpie pen because you obviously can't see it. So I'm going to show you how to streak out on a plate single colonies. And what's important about single colonies? The reason why you would make single colonies is to make sure that the strain or like the bacteria or the yeast that you're using is pure. Now, if you streak it out very like lightly or like very little cells, then it'll actually make individual tiny colonies. And you can tell that they're pure colonies by checking out this thing called a morphology. And the morphology can be anything from the size, the color, the texture, and even like the number of colonies. And if they all kind of look the same, they look very similar, at least you can say, okay, I have one species of X here. And yeah, that would be a good idea to check and make sure that nothing got contaminated. So let me show you how to streak out very professionally with a Sharpie pen on this new plate. So yeah, you're going to open it. I'm not going to open it. But what you're going to do is you're going to just dip your inoculation into the bacteria, and then you're going to spread it very densely all on one side. Now you're going to take this loop and you're going to throw it away. Once you throw it away, you grab a brand new loop, grabbing a brand new Sharpie, brand new loop that's not been used yet, and you take a very small area of this and you pull it out. So pretty much a dense amount of cells and maybe a little less dense amount of cells. And you take this and you pull it out and then you streak it like this. And then with that same inoculation loop, you throw it away. And then you get a new one. You get a new inoculation loop that you haven't used before. And with that, you take a very small amount of the end of that. You pull it and you just loop it out real nice without touching it. So as you can see, you kind of have this little dense area, a less dense area, and then you finally streak it out to potentially single colonies. Now, if you couldn't do that, that means you have way too many cells to begin with. Yeah, yeah. Any questions? Shiv asks, can you boil these loops and reuse them for later? Can't really boil plastic. Would not recommend it. But I have seen people take the loops and sanitize them in 10% bleach. And then once you sanitize them, I would use some water and just kind of like give them a little shake just to make sure you don't put 10% bleach right directly into bacteria. So yeah, if you're like dying to use a few loops again, you can definitely use 10% bleach and sanitize it all. But they're generally like pretty cheap. You can get like 100 for like $5. So, okay. Generally, we need to keep the bacteria alive. So we do only send you 4 stabs, and this has to last you about 2 more months. So what I recommend doing is right when you get your stab, you streak it out on a plate. And then a month later, you're going to take 1 colony of this, one little dot, and you're going to re-streak it on another plate. And this will ensure that you have something consistently growing, because once your first plate after E. coli grows, then eventually it kind of like outgrows itself, and then it eats all the media on this, all the nutrients, and then it eventually just like eats itself and then dies. The plates sometimes like even shrivel because, it's exposed to air and things like that. So they will shrivel and become like this little thin plastic. I'm sure some of you will see it. So for BBOSW0001, so for E. coli, what are we looking for to determine whether we have a single colony? So after this whole method that I did, hopefully in this like region where it's less cells, you can have, it looks like a circle. It looks like a tiny little circle. That means it's 1 colony of bacteria. And that means it's all uniform. Usually you see like just blobs of streaks, and that's not essentially what we're looking for. We're looking for just a round circle. And same goes for yeast. They just look a little different. Bacteria and yeast look quite different, as you'll see an experiment with. One is very small in morphology, and one is very shiny. So lots of things can determine if you have a pure strain or not. Demonstration. So once you streak out the plates, the way you're going to grow E. coli or bacteria or yeast is to put them in the proper temperature, right? Because they need a really good amount of temperature to grow. So we usually keep out the plate at room temperature inside of our house. And if you live in a colder climate, you can keep it outside. You can keep it out if it's like 57 degrees Fahrenheit or like 40. It'll just take a little bit longer to grow. The optimum temperature for E. coli is
36, 37 degrees Celsius.
36, 37 degrees Celsius, which is like 75 to like 80 degrees Fahrenheit. I'm really good at maths. But like like I said, it doesn't matter too much. Just the amount of time that it'll take to grow will vary because dependent on temperature. If it's colder, it's going to take longer. If it's hotter, it's going to grow really fast. Temperature fluctuations, Matt, is not essentially bad for the colonies. It's either they grow or they don't. Like they don't mutate or anything under certain temperatures, so I think you'll be fine. BBOSW0001 has calculated and it's 97.7 degrees Fahrenheit. Whoops, no, not that hot. What is it? It's like 30.
33, I guess?
33, maybe? Yeah, room temperature. Room temperature.
Room temperature is 27. Yeah.
37 is human body temp. I guess you can like put under your armpit and just kind of grow it in your armpit, but that's not cool. Yeah. So yeah, once those nice colonies form after a few days, and there's lots of little colonies, then what you can do is you can take it and put it in the fridge. Put it at -4 degrees Celsius, and you will keep it from going bad. Yeah. That sounds good. So once you finish engineering, and you know all the plates that you've done thus far, let's say all those hand washing plates, I'm sure they turned out really gross. I saw a few bath mats. What did I see? So there was a bath mat, there was kombucha, there was like tons of gross stuff you guys are growing, which is fun. And I'm sure you guys found a lot of different things. So what you can do with those plates now is you can just throw them away. They're not harmful to you. They won't like fester and kill everybody in your family. You just throw them away. With bacteria that you might handle in liquid form, like in a broth, let's say, like liquid form, you can just put 10% bleach inside of it and literally just swirl it around and it'll fizz up and all the bacteria will just be like, and eventually just die. So you could just throw it in the toilet. Not a problem. And yeah, any more questions before we sign off? Yes, sub-3, your athlete's foot will devastate everything. Okay. So I look forward to what some of your good colonies might look like. Make sure you post it in our Facebook group. I will go and critique every single one of them, which would be really, really cool. Chris asked, the glowy bacteria part of the second month's package? Yes. Oh, I forgot to mention. Yeah, the bioluminescent bacteria kit is included in month 2. Make sure you read all the protocols and then try and kind of tackle it on your own. I'm going to give a demonstration later of what it's supposed to look like. I won't give it now because I want you to struggle with it because I'm a cruel person. Pete Gill just asked, how does antibiotics help with contamination? Ooh, that's a great question. So we sent you a lot of LB agar, and LB just contains just the minimal amount of things that, or I'm sorry, not the minimal, pretty much all the things that bacteria need to grow and flourish on. So if you don't add an antibiotic to just the regular LB, you can see that a lot of other things will contaminate, like the air and yeah, your mouth, like anything that comes in contact with the plate will flourish and grow. Now, if you have the addition of antibiotics, let's say streptomycin and kanamycin, then things will become selective and things that you want to grow on it will grow on it. Things that you don't want to grow on it will not grow on it. So antibiotics are good fail-safe to use in the laboratory and to create your plates with. Mike D just drinks the broth, helps the immune system. Gross. M. Corel, yeah, so with the colonies thus far that you've made with all those like handprints and kind of tested out like how agar works, you can throw it out or you can keep it as a pet. Put some googly eyes on it. I don't know. I weirdly saved a bunch of plates that had really weird growths, and yeah, it eventually got to the rest of my experiments. Like it started spreading to my other experiments. So my boss told me to throw them away. And I think I had about like 50 different really cute plates with googly eyes on them that had like furry things growing inside of them. But that's just me. Maya Minder asks, can I use blood agar plates? I'm not exactly sure what blood agar is used for. For the practicality of this course, we're using just regular LB and YPD plates, also supplemented with lots of antibiotics. So unsure about blood plates. I think you might use them for like human or mammalian. But yeah, for the sake of this course, we don't use any of those plates. Blood. All right, cool. If there's no more questions,